Okay, I guess you can see the presentation now. Uh, well, first I want to thank you guys from the Shark Trust for inviting us and having us in this really awesome event. It would be great to see so many scientists talking about angel sharks from all around the world. And well, I would say that I'm delighted to be here. And well, my name is Francisco Concha, also known as Pancho for my friends. And you may not know me yet. So I will tell you I'm from Chile and I'm the PI from Condola. And I would say that, sorry, um, here's where I work. So I wish the whole building were my lab, but it's not. It's just one of the labs in here. So we are in central Chile, in the region of Valparaíso, and um, so here, this is Universidad de Valparaíso, and well, this is the people in the lab. So this is I'm the PI for Control Lab with my wife, and you can see here some of the students, some other colleagues now. We work in different areas, but mostly on shark biology and skate biology conditions. This is Rafael. This is going to be the future PI. And so one of the things we do in our lab is biology, of course, and we aim to work in conservation too, but there's no conservation, there's no biology if you don't work first in the identification of the species, right? So one of the things one of the areas we will try to cover in here and in our lab is taxonomy, actually. Not my favorite area, but I would say is the most important one because nothing can be done if you don't have a real and a reliable ID of your specimens, right? So we are involved with in a project of University of Connecticut and University of Kansas. This is a huge project in the U.S. So I'm one of the hundreds of students that like great students that have been working in there. So I'm one of those. And but we also in our lab here in Condor Lab we have this, as I told you, we have this taxonomic area. So with one of the students we have in the lab, we started checking what was the status, the taxonomic status of the species we have of conjunctions we have in Chile, and it was very interesting because well, thanks thanks to Carlos Bustamante, so because he did the hard work of checking all the literature, and in 2014 he provided us this really cool list of species reported for Chile. So he reported about 103 species of conjunctions, and so ever since then I think we added like we need to add like three more species. So we have about 106 species of conjunctions in Chile. And so one of the things we were trying to do with our student was to see what was the taxonomic status, which means not only which species do we have in Chile, but also which of these species were actually described in Chile. So how many Chilean species do we have? So in the end, he checked all the literature. We checked the linear descriptions and stuff, and then we got to these numbers of 17, this number of 17 species from the Chilean coast. So describe it from Chile over all this time. So one of the main issues we have in Chile is that in the Southeastern Pacific, I would start saying it as the Southeastern Pacific because it's not only a Chilean issue here. So we have the same thing in Peru, maybe in Ecuador, and also in part in Colombia. So we have that most of the species we have reported in here, those are classified as data deficient by the red list of the International Union of Conservation of Nature. And if they're not data efficient, they will be not evaluated. So I would say we in Chile we are pretty much in the left side of this table. So we have there are really few species that we actually have enough information to do a proper assessment, right? So it's been I would say it's bad news for contrictants, but it's pretty good news for us because then there's a lot of things we have to do now. So giving that context, then I would say that when we start working in taxonomy, we want to ask some questions, right? So the basic three questions we ask about every single species that we know now that are described in Chile was, so what's the quality of the description? So the first question is, how is that description, or how was it made? Is it complete? Is it incomplete? Is it large, short, etc.? So that will be the first step. So check the description, check the quality of the description, and compare it to the description that we can find actually in these days, right? Because now they're much more complete. They include much more measurements, and they include other stuff, right? And the other thing is, what was happening with what about the whereabouts of the type material? Because you can have a really good description, but then when you go and check in the script in the same publication, if you want to see where the specimen is now, where the holotype is, you'll be surprised, but in many original descriptions, there's actually no mention to where the holotype is. So in some of them, others you really have to guess and check other publications. So it's kind of hard to track back. And so the whereabouts of type materials, that is a very complicated thing. So especially in a country like this, where we have all kinds of natural disasters, like every 20 years we have a huge earthquake that would destroy everything, or the fires or pirates in the past, and I don't know, we're waiting for Godzilla or something like that. And now, so we check this, and most of the species that we're actually describing in Chile, they don't have type material available in the museum in Chile where they're supposed to be. So most of them went to the British Museum, or they went to Harvard, and some of them stayed in the museum. But of those that were actually kept in the museum in Santiago, most of them were lost in a fire. So, and some other collections, well, they have the same fate. And what about molecular data? Well, as you know, molecular data started to be taken like really recently. So most, of course, none of the old descriptions will include molecular data. So you're not able to compare morphologically specimens from this side of the world with others from other places. And of course, molecular data will give you a lot of help when you are checking conspecificity, when you have broad distributions, right? So many cryptic species have been discovered just because molecules have ring the alarm. And in this case, for the species we have in Chile, that most of them were described in the 1800s, it's been really hard, and there's not much material to compare with other places. So having all this in mind, and considering all what I just said, so one of the first genera that, because we start working in several genera, we have a couple of students working in different skates mostly, but one of those genera that were of interest was the angel sharks, right? So André Marie Constant Dumeril, back in the 1800s, he described it in the genus Spadina, and his description, when I see his picture, I cannot stop thinking that he looks like a DJ. So I will do this. Okay, so the description was really short and brief, but I would say it was fair enough. I mean, in those years, there wasn't much to see. I mean, that was the way things were done, right? So there are many genera that were actually really briefly described. And I would say, well, the description of this genus, so Squatina, also known as the angel, this is what he mentioned in the original description, has fins notched at their origins, the mouth is in the end of the head, and the head is rounded. So yeah, we can say that he kind of missed a couple of details. But if you see the description, I mean, yeah, there's not much you can, when you see that the fins are notched in their origins, well, that reduces the list immediately. So I would say it's super brief. Total probably is not enough to have a really good idea of how it looks, how they look like, but still they it's still a good description for a genus, considering that there is a diagnostic trait that is actually helpful in this case. So then we see, okay, we have Squatina. So one of the Squatinas we have in Chile, or that were reported for Chile historically, was Squatina californica, and that species, I'm using the distribution proposed by Compaño and collaborators in 2000, back in 2005. But I would say historically, the species was considered to happen in Chile. It was reported in Chile for a long time, and nobody questioned that idea for a long time, I would say, and until, well, suddenly we start looking at the specimens and say, okay, how 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 do we know that this is actually Squatina californica and it's not the other one? And actually, we didn't have a good answer for that question, because it's really hard to tell them apart if you don't have them both together on the table to check them, right? Because if you see the descriptions, well, you will see that this species, Californica, was described by Ayers in 1859, and was described from San Francisco, California. That's where the name comes from, right? And his description was brief, not super brief by the standards for those years, but it was kind of brief and had a lot of details in terms of morphology, but it didn't have any measurements, or it didn't have many numbers in it. So you didn't have comparisons of how was the I don't know, the size of the orbits with respect to interorbital space or the spiracles, that kind of comparisons you have now. So, and they didn't provide a picture, of course, 1859. So they used to, authors in those years, they used to include a diagram, right? So a diagram that actually shows the most important traits, so you can recognize this. And also the other thing is that in the original description of Spadina californica, he described it and also compared it to Spadina dumeri, which was the one in the other side of the US, in Atlantic. So there was no other comparison with other species by described by them, only that one in the other side. And because I guess there was not there was another species in the Pacific, in the Eastern Pacific by then. So, and the drawing that he included in there, so the scheme was this. And yeah, it looks, I'm not criticizing the quality of the drawings, because I'm awful at drawing, but I would say, yeah, I would say this is an angel shark. Of course, is this drawing useful to actually identify the specimen from other angel sharks? Not sure. So later, back in 2005, as I mentioned before, so Compaño collaborators did this Sharks of the World, and that was an interesting book because, well, we all want one book with all the species in it, so you can actually see all the drawings and stuff and descriptions and remarks, right? So that was it was a huge work, following up a little bit what he did in 1984, right? And he included a diagram of this case. So I would say the drawing was a little bit better in terms of, it looks more detailed. You can see some more details in the spiracles, you can see more, well, the shape of the fins, the pectoral fins is pretty much the same, the other is pretty much the same. So I would say at least the shape of the head is a little bit more detailed. And also, well, you can see the differences in the braid. But there's this thing in here that I'm not very sure about. It looks like he's showing some kind of thorns. These thorns are not mentioned at all in the lineal description. So this might lead you to confusion. So, but how does Squatina californica looks like actually is that is the species. So well, I look for a bunch of pictures of the species. So this is more reliable I found, considering how hard this is to ID this guy. So I found this, and as you can see, if you put both drawings together, maybe you can make a real Squatina californica. But yeah, the drawings are not telling you much more about coloration patterns. There are these weird lines in the middle which resemble spinulation, but they actually don't have that. And there's nothing about other spinulation and other patterns you can see here. So I would say in this case, the drawings might not be very useful to at least to distinguish between squatinas from the same side, right? I mean, I won't criticize Mark Dando's work because it's awesome, but I would say for the sake of identification, this might not be enough in this case, right? So, well, this is Squatina californica. You can see it more in detail. So you have this pattern of coloration. You have these white dots that they look darker when the specimen is fresh. I guess this is a preserved specimen. So of course you lose some coloration in that, but then you can compare it to Dando's drawing, and I would say it's pretty accurate in terms of general shape. So well, it is what it is, and it's fine. But so remember, so Californica was described here in San Francisco and was reported, and the distribution proposed by Compagno based on reports and etc. So it was from here, from San Francisco, well, California to Baja California, and then there was nothing in between it. And then we have it again in Ecuador, Peru, and Chile. So that is interesting because you have a distribution that is actually not continuous, which is kind of weird. But well, those I guess he picked up the most reliable reports, and he decided what to include. So, and the other species we have in Chile was Squatina armata. So this is our case, and well, it was really cool because this guy here, so Rodolfo Amando Filippi, he's a zoologist. He was a zoologist, and he worked in the National History Museum in Chile, and he described a bunch of species. And he was very, I would say, he did a very good job in describing the species. He described other conditions, right? And but so here's the drawing he included, and it was actually, again, so in those years you had some kind of artistic view. So most of the scientists were not doing the drawings. I mean, not all of the scientists were like Haeckel, for example. So they hired some artists, and the artist was including things that he imagined, right? So we got this drawing for the to go with the description. And well, the type locality was in here in northern Chile, so in the very north, in the city of Iquique. And well, here's the drawing again. So you can also see this band in here in the middle, which I'm not sure if it's an effect of the copy, like a printing problem, or it's actually showing some thorns, right? And that was very confusing, I guess, because, well, as you can see here, again in Compagno 2005, you can see that he drew here two lines or two rows of medial dorsal thorns and caudal thorns in here. So you can see that band in here, which may or may not be actually thorns. And then you have this drawing with thorns, clearly thorns in here. But if you see the real species, right, like a fresh specimens, you won't see those in here. So you will see the spines in here, you will see the spines in here, you will have a lot of thorns in here in the head, in the forehead. And those are described by Filippi. Filippi did a really good job in describing the species. The description was really complete and extensive. It was lacking some comparisons, of course, that some of the ones that we do actually nowadays. But the description in general terms was pretty good. So now we have this Squatina armata and Californica. So Californica goes from here up to here, and then we have the distribution proposed for Spadina armata was from exclusively from South America. Doubt they doubt that it was actually here, but you have a really solid line here in for Chile actually. So the question here, well, we have a lot of questions, right? And how are we sure that these both species are actually sympatric in here? Are they here only here, or they only or they also share the distribution in Peru and Ecuador, so in Colombia? And so in order to do that well, we need to check the ID. So we decided that's why we decided to pick up this specific project like Squatina armata with these colleagues, because it was actually a problem. We didn't know, we were not certain of what do we have in here in Chile. So first place to look for, we went to the website for the red list, and then we saw that, of course, surprise, the species is data deficient. This was an assessment by Julio Lamilla and Miguel Romero in 2004. So now it's going to be updated really soon, and it might change or not. I don't remember exactly, but I'm pretty sure there should be more information that it was by then. So we checked literature, and if you do a quick search for this species, for Squatina armata, then you will find some parasitology. So the Castros, he reported some copepods or some ectoparasites. Then you have this great dissertation by Abioro, which is about systematics, and you have this report from Hernandez and collaborators about reports of bycatch. So this species being bycatch of different fisheries. And Viares et al. From, which is pretty much, it's archaeology. So he reported, these authors reported some remains of Squatina in some, in different parts in north of Chile, like remains from Indian deposits. So basically everything is about distribution and reports of the species, of the presence of the species in different places, but nothing about actually discussing the ID, or at least putting it into question. So the taxonomy, so what are we still missing? So taxonomy first, that's the first thing we're missing, and that's the first thing we need to do in order to get all these other blanks filled. And then what should we do? Well, what should we know? We need to know how is the description. Well, we checked the description. The description of Spadina armata was pretty good, but it needs to be done again in terms of to add some comparisons, some measurements, right? Then you have type material. Well, what about type material? Type material was deposited in here in the museum in Santiago, but it was loaned to Universidad Austral, so to southern Chile, to another collection, and there was a fire, of course, and then the specimen got lost. So that's really sad, but happens all the time. So what do we need to do in order to get new material? So new type material. So we need to go to the type locality, to Iquique, remember? So we need to go to there, and once we go there and get the samples, we will be able to deposit new type material. We can do a neotype, we can establish a neotype, and then we can redescribe it. And of course, we can take molecular data and do the things like the way they get done now. We were super lucky, and in a couple of weeks, I was talking with friends in that area, and in a couple of weeks, we got actually 2 really nice specimens, big specimens, more than 1.2 meters or so adult males. And so this is the team. So this is Miguel Araya, my colleague from Iquique. So he was the guy that actually collected the specimens. And this is Italo, and this is Sebastian. So these are colleagues from other universities here in Chile. So both are from Chile, and they were really enthusiastic in doing this project with Squatina. So actually, I think it was their idea, I would say. So we start doing this. Once we collected the specimen, we got this really beautiful male and another one. So we will have a neotype finally. It was super awesome. So now that we have the specimens actually in our hands and we put it in the collection, we were really ready to go. So we have the material that's done. Now we're ready to take, well, molecular data. We can do DNA. You know what these genetic guys do. And oh, by the way, when we do X-rays, when we did the X-rays, we actually saw that Squatina eats fishes. Well, not surprised, but at least now we have a data, so now we know what they eat. So we got the specimens again, and we will be analyzing all spinulation, size. We'll measure everything, every single trait, and we have a bunch of really cool pictures to add to the description. And of course, we will go more into detail than it was done originally, because now we have more species to compare to, right? And now we know there's actually an issue with the description of these species and the comparison we need to do with the species from the north, right, in California, because we have to make sure that what we have in here is armata and not California, or if we have both, right? So at least we have it from type locality now, so we are pretty sure, because the description was consistent with the morphology of the specimen we collected. So now we know this is a real armata, and now we have to wait to see what happens with Spadina californica. But that's another issue, right? It's not my problem. Well, kind of is, but I'm not going to solve it, because that's something that people should do in the US. So this is a spinulation of the pectoral fins, which is what gave the name for the species, right? The species was called armata because of the many spines it has, so it was armed. Here's the X-ray, so we can do meristics that were not done by then. So in the original description, you don't find the counts of vertebrae, for example. We will have counts of pectoral radial, pelvic radial, so vertebrae, I said that already, so we will have the spines and stuff, so we can do morphology comparisons and also meristics. That's what we are going to add. We will be able to count teeth, actually, which is super awesome. And not only that, so in the original description, clusters were not very well described. So now we can include that, even external morphology. Then we can talk about the skeleton of the clusters. We actually have two specimens, so we can leave the paratype untouched, and then we can just cut the other guy and have the skeleton made. We did this with the help of one of my students in the lab. So we have the skeleton, and we'll label all this in the description, right? So to conclude and to finish, we will say that what happened with the description, well, we know the description was pretty good again, but we will do it more complete, and we will update the way that it was described in a new redescription, which is needed because now we need to set up, we need to establish a neotype in here. So type material, of course, the holotype was lost. So we will set a new type, so a neotype, and that will fix the issue. That will totally fix the issue. So now we will have something to compare to other species in other specimens. We will get, and actually we have sequence for these guys. So we have molecular data, we have ND2, and we have CO1. So just to have to be able to compare with more specimens in other places. So we have molecular data that's done. So grateful, and we were so lucky that we did this before the pandemic, because now everything is banned. You cannot go in your lab, and there's nothing we can do about it in Chile for a while. So we're lucky that we got the molecular data already done. So that's super great. And in collection, well, now we will be able to set up a collection and tissue collection and also specimen collection. So now the thing, I would say the circle is kind of closed now, because we will have a redescription. We are making it, of course, it's not finished yet. We are doing the redescription. We are putting material in a neotype in the museum where it belongs. And we have molecular data, so you can compare it to your molecular data anywhere in the world. And we actually will have a collection which will include tissues in the museum. So that will be awesome, and that will be super helpful to answer our question. So do we have Squatina armata, and do we have the other? So that's what I have to say. Now there's the issue of Squatina californica. That will be a problem now in the north, I would say, in Peru, Ecuador. They have to see. I've been there, and I've seen what they have. and they may or may not have it. So now that we have all this data, now we will be able to compare to specimens they have collected. So we will finally be able to say whether they have armata or not. Whether they have Californica or not, that's a different issue, because to do that, in order to do that, we need to check this, the type material for the species, which is supposed to be in California. And I think it's lost. So I think that the holotype of Squatina californica is lost. So I think it needs for redescription, it needs to be collected again, and a neotype needs to be put in the collection. So pretty much the same thing we are doing here, they need to do it in San Francisco. So that's what I have to say, and that's the project. And well, we are aiming to finish this as soon as possible. At least we have the specimens, we have all the measurements done, and we are just writing. So I hope we will have news very soon. So thank you for listening to us and for letting us tell you what we're doing here back down in Chile. And I want to thank all the other co-authors in this work and the hard work they have been doing. And of course, well, I will invite you to contact us in case you have any inquiries. And there won't be a way that you can ask me directly now, but at least here's my email, and this is our website and our social networks and stuff. So you can contact us, and if you have any further questions or there's anything you want to do here in Chile, well, there is people in Chile that is actually working in constrictance now, in constrictance biology, so you're not alone anymore in here. So thank you very much, and I will see you guys later. Thank you.